Our next speaker is Fernando Baquero from Madrid, Ramon y Cajal Hospital. And Alyssa has asked me to remind the speakers that we are videotaping. So first, if you prefer not to be videotaped, please let us know. And secondly, please speak into the microphone so that they can record your words. Do you have some pictures from outside somewhere? Yeah. I think we're going to enable it.
Thank you, my friends. Thank you, Mark, for inviting me. Thank you, Eliza, for organizing everything so good. So I will try to make a relatively provocative presentation about my feelings about the possibility of using a new concept, the possibility of using Eco-Evo drugs to control antibiotic resistance. Let's start by the classic approaches to contain antibiotic resistance. As it's defined here, you can see on the left the resistant organism, a clone, a pathogen, which is entering in a host in yellow, and penetrating in that host is causing infection. And then this bug, this resistant organism or clone or pathogen may spread to other patients and may perpetuate himself even in the same patient, for instance, as a carrier. So a number of things has been proposed to contain organisms, resistant organisms, including obviously sanitation, vaccination of the host in order to prevent penetration, search and destroy strategy, treating early and potently to eradicate the pathogen, making containment measures just to try to prevent host-to-host transmission, and looking in general at antibiotic policy, how to use antibiotics to contain the selection of antibiotic-resistant organisms. So my point today is that all that is the classic thing, but that's not sufficient anymore. The efficacy of these classic methods is inversely proportional to the density and penetration of the resistant organisms and the resistant mobile genetic elements. So if we have many resistant organisms and many resistant genetic elements, these type of approaches are less interesting than we hope. So useful measures at early stages of resistance in clean areas have no effect in areas where resistance heavily pollutes the environment. So we can control antibiotic resistance in the Netherlands, but with a big difficulty in India or in Thailand. So in these areas, densely polluted with antibiotic resistance will invade sooner or later with resistance the clean environment. So the idea is that we should approach something bigger than the hospital or the patient in the middle. We can act on the patient to try to cure better the patient and to prevent transmission from this patient to other patients. We can act with antibiotic policies in the hospital, but the problem is that antibiotic resistance is spreading more and more into the community, and not only that, in the environment. So we have now not only a hospital problem, but an environmental problem at large. That's what I call planet medicine in general. So putting a very descriptive figures, antibiotic resistance is leaking from classic containment measures. Antibiotic resistance is spreading all around in the environment and is just out of control of the measures that we can do at the hospital. That's why we were adapting the concept of Eco-Evo to the antibiotic resistance. That means not just looking at antibiotic resistance bacteria as target of, as causative of infections, but broadly in the light of its evolution and ecology. So to control that at this level, we need to change our mind about what is a drug. And we should change our mind from drugs to cure patients to drugs to cure sick environments, because it's the environment which is sick of resistance. It's not just the patient. It's the hospital which is sick. It's the community which is sick. It's the river which is sick. It's the farm which is sick. So we need probably drugs and interventions that aims not necessarily to kill resistant organisms, but also to prevent their emergence, spread, maintenance, and evolution, and eventually to re-establish in bio-restorative attitudes to re-establish the antibiotic-susceptible populations. Part of what I am saying today is published in a mini review that I I published in Antimicrobial Agents and Chemotherapy last August, so if you can look at that. So anything that matters in terms of the spread of antibiotic resistance at any level depends on the type of biological units that spreads resistance. That may be from clones to genes. And all these structures spreading antibiotic resistance have a number of properties that are common for all of them. All of them penetrate. It's an ecological invasiveness, transmissibility. Many of them are promiscuous. They connect with other antibiotic resistance platforms or genes. They are plastic. They are able to have variants, to recombine, to be modularized. And they persist. They have ecological persistence during long. So that's a schema on the 4 Ps. The universal processes that occurs for any type of resistance unit in biology. All these units should penetrate, be promiscuous, be plastic, and persist. And that's valid across the genetical hierarchy, from metagenomes to species, to clones, to mobile genetic elements, genes, or modular genetic sequences. So we have the classic thing, a resistant organism, a pathogen is penetrating in a host, and the host is spreading this organism to other hosts. But note that the same occurs with a resistant gene. The resistant gene is penetrating in a genetic platform, an integron, for instance, and the integron is able to penetrate in other genetic platforms, provoking a variety of genetic platforms, for instance, different integrons. And the same occurs with these genetic platforms that penetrates in a plasmid or in an integrative conjugative element, and this plasmid or ICE is able to spread their genetic platforms in other plasmids and distributing them the unit itself. So penetration and promiscuity occurs all around. The resistant plasmid should penetrate in a bacterial host, and the bacteria should succeed to infect a host, and this host has, as a patient, has the possibility of spreading to other patient. This bacterial host has the ability to transmit the resistance plasmid to many other clones or bacterial hosts. Finally, the resistant clone penetrates in the host, and the host distributed it. So at the end, we have the same phenomenon for penetration and promiscuity for all type of levels of genetic levels of diffusion of antibiotic resistance. Plasticity also occurs for everything. Genes and platforms and clones are able to mutate, to recombine and modularize, producing also the possibility of changing, continuous change in the structure of these elements. And persistence is quite important. To be important in antibiotic resistance, you should persist during long periods in the same host, in the upper part, or persist in different environments, in different hosts, but persist. That's, for instance, what happens in my hospital with this red clone, which is the ST18, is sequence type 18 inside a very particular clonal complex 17. Well, you see that this exactly the same clone inside the particular clonal complex of Enterococcus faecium is able to produce during decades bacteremic episodes among our patients. To a certain extent, this clone is established in my hospital, has perpetuated himself in my hospital. He is in certain extent domesticated by the hospital. So, of course, all that is related, and that's a Russian doll system or something like that. One thing is inside the other. That's a multi-level biology of antibiotic resistance from the gene to the environment. and all that at different levels, and all these levels, I think, should be attacked simultaneously. I will not enter into the details, but that happens in clinics, apart from the, let's say, speculative nature of this talk. I am doing real science in my lab, I should assure you, and that's based on real observations that we did. On the left-hand side, we can see clinics that have a very long penetration in the hospital during years, and then have plasmids, and plasmids are transferred to other organisms, and these organisms share genetic platforms, and these genetic platforms are able to spread particular resistant genes, like in this particular case, VIM1, a new carbapenemase. The penetration of anything from a gene, from a clone, from a species, from plasmid in the hospital facilitates promiscuity, because if you you penetrate many environments, you have contacts with many other people. Promiscuity favors plasticity, and plasticity favors persistence, and persistence penetration, and so on. You have circles and circles in which you can act upon a certain moment. It's a restricted field for useful intervention, just limited to a reduced number of interactions. So I think we should more and more be able to diagnose the sick environment, so making surveillance of high-risk associations between species or clones with particular plasmid. If a particular invading species is associated with a particular dangerous plasmid, able to attract transposons which carry particular resistance genes, or carry integrons which are able to recruit novel resistance genes, we can diagnose an association which can be eventually be treated. And in fact, what we are doing all the time in my lab, in other labs in the world, is doing this type of multi-level epidemiology, which is becoming our daily task in our lab. That's some publication of the last years of my lab, looking simultaneously at beta-lactamases, genes, clones, plasmids, transposons, integrons in the same location, in the same hospital, just to try to understand the total plot, what's happening. So come to that, we need a change about what's drug, from cured patients to cure environments, and what to do to cure environments. With few exceptions, there's a very small number of promising products available, and there's no experience in the Eco-Evo strategy in real world, and of course no regulatory guidelines, which is a problem. But I will, in any case, make some words about what's the possible perspectives for this approach, which is essentially based on the 4 Ps. The first P is inhibiting the penetration. Inhibiting the high-risk clonal complexes, I think, is absolutely critical. In many instances, the rise and fall of antibiotic resistance depends more on the invasion of a particular clone which has a particular mechanism of resistance than from any other thing. And the number of clones that are spreading with mechanisms of resistance are relatively limited. But the resistance goes up and down, not because of the antibiotic use, in part of the antibiotic use, but in many instances because of the dynamics of the clone itself. So controlling high-risk clonal complexes are critical, and we need probably focus, for instance, approaches to treat that as particular vaccines directed to particularly high-risk clones. And everybody knows the type of clones I'm speaking about, 256 in Strep pneumo, the 8 in Staphylococcus aureus, the 131 in Staphylococcus in E. coli, the ST17 in Enterococcus faecium, the ST2 and Enterococcus faecalis. A number of clones that are spreading all around the world, antibiotic resistance. We need probably vaccines against that, or drugs trying to decrease their ability to penetrate, targeting surface antigens, quorum sensing, and therefore colonization possibilities. Or we can try to reduce selective environments, that if these clones goes into a particular patient, don't to select them effectively. And there's a number of approaches trying to reduce the selective activity of antibiotics on the microbiota. For instance, deactivating active antibiotics in the gut by using local oral beta-lactamases that destroys the selective agent in the gut, or antibiotic binding substances. We can have also promiscuity inhibitors. That's extremely important, because that's the possibility of trying to interact or interrupt the traffic of plasmids between bacterial populations. Of course, we will not treat every plasmid, but just in these occasions in which we have localized a particular patient with a particular clone that has a plasmid that we would not like to be this plasmid distributed to other organisms in the hospital. We can eventually think in the future to apply broad host range conjugation inhibitors, particularly type 4 secretion system inhibitors that are inhibitors of the mechanism by which the plasmid are transferred from one cell to the other, including the relaxase that nicks the DNA before transmitting, and the system of ATPases which are needed to provide the energy to produce the system. So there's a number of type 4 secretion system inhibitors that are working. The clodronate or etidronate, there's something that is start to be proved to act, at least in microcosms in vitro, as relaxase inhibitors impeding plasmid transmission. We can think about specific conjugation inhibitors. The first one, the broad host range conjugation inhibitors, is interesting because this type 4 secretion system inhibitors is very widespread in many conjugative plasmids and integrative conjugative elements. Also more specific may be type 4B pill inhibitors, or using inhibitors based on CRISPs, CRISPRs, just using that as an anti-plasmid strategy. So that's to remember that there's common mechanisms of conjugation. There's an ubiquity of these systems, including these relaxases and the systems for transfer of the single strand to donor to recipient cell. Plasticity inhibitors, something has been presented in that field in the previous talk. There's a possibility of targeting RIC-A, targeting integrases. We have a lot of integrases that are just put on the basket that were used for inhibition of HIV that can be eventually reused to discover how to prevent integration of resistance genes in integrons. Mutation inhibitors, something that has been also addressed in the previous talk, targeting SOS gene networks, and therefore reducing the ability of these systems to provoke mutations. Finally, persistence inhibitors, decontamination of high-risk clones. That's something difficult to do, but eventually we can target clones by anticlonal vaccines in order to decontaminate populations for particular clones. And in fact, something like that has been successful in the case of the Streptococcus pneumoniae conjugate vaccine. That has decontaminated part of the population of a number of Streptococcus pneumoniae clones. That is indeed the most successful anti-resistance approach that has been ever established. We can try to target colonization and virulence of these high-risk clones, or just decontaminate. Plasmid decontamination is extremely difficult, as you can imagine, but there's a number of compounds that can be used for that. Plumbagin, for instance, and other compounds has been started to be used, at least in experimental models. We can use theoretically to decontaminate mobile genetic elements like plasmids, phenomenon like incompatibility of the phenomenon of toxin-antitoxin systems in plasmid. Decontamination of high-risk genes, I think, is out of the possibilities, but theoretically there are tools for doing that. So that's all I wanted to say, except my acknowledgements and a couple of slides more. So first to mention that I just edited very, I think, interesting thematic issue of the FEM's microbiological reviews on antimicrobial resistance, in which we collected a number of papers just related with the type of thoughts that I was trying to provide this morning to you, and how antibiotic resistance should be simultaneously considered from many levels, from the ecosystems to the genes and along the hierarchy. That's essentially a gene flow, this soup of plasmids that is converted in dangerous high-risk clones. And all that had been impossible without the pioneering work that I would like to honor in my last slide, which is the 50 years this year. We commemorate the publication of the first paper about plasmid transfer of antibiotic resistance in pathogenic bacteria, in Enterobacteriaceae, by Tsutomu Watanabe and Toshio Furusawa. And so that's my last slide honoring these people that started the field. Thank you very much.